Good morning, class. So, today the topic is histology of salivary glands, and in this topic, we are going to cover the composition of salivary gland, anatomy, development, and structure of the salivary glands. Starting with the introduction, salivary gland, exocrine gland whose secretion flows into the oral cavity. Three pairs of large glands are located in our oral cavity, extraorally, and they are the major salivary glands. And many minor salivary glands are located widely. Major and minor salivary glands are composed of parenchymal elements and supported by connective tissue. Parenchymal elements are derived from the oral epithelium and consists of terminal secretory units leading into ducts that eventually open into the oral cavity. Connective tissue forms a capsule around the gland and extends into it, dividing groups of secretory units and ducts into lobes and lobules. Production of saliva is the most important function of salivary glands. Blood, lymph vessels, and nerve that supply the gland are present in the connective tissue. Saliva contains various organic and inorganic substances, and it provides the natural protection for teeth and soft tissues of the oral cavity that helps in mastication, deglutition, and digestion of food. That is chewing, swallowing, and digestion of the food. The major salivary glands are parotid, submandibular, and sublingual salivary glands. Numerous minor salivary glands are located in various parts of the oral cavity. They are the labial, lingual, palatal, buccal, grossopalatine, and are typically located in the submucosal layer. To summarize the composition of the saliva, the volume of the saliva secreted per day is about 600 to 1000 ml. The electrolytes present in saliva are sodium, potassium, chloride, bicarbonate, calcium, magnesium, fluoride, and so on. Secretory proteins that are present are amylase, proline-rich proteins, mucin, histatin, cystatin, peroxidase, lysozyme, and lactoferrin. The immunoglobulins are secretory immunoglobulin A, immunoglobulin G, and M. Small organic molecules like glucose, amino acids, urea, uric acid, and lipids are also present. Other components include insulin, cyclic adenosine monophosphate binding proteins, and serum albumin. The flow rate of parotid and submandibular gland at resting and when stimulated are given as following. Saliva produced differs in amount and composition in each salivary gland. Parotid gland secretes watery saliva, which is rich in enzymes such as amylase, proteins such as proline-rich proteins, and other glycoproteins. Whereas the submandibular gland produces saliva. In addition to the components which are present in the parotid gland, it also contains highly glycosylated substances called mucins. Sublingual gland also produces a viscous saliva, which is rich in mucins. Coming to the anatomy of the salivary gland, the parotid is the largest salivary gland, and its superficial portion is located subcutaneously in front of the external ear, and in this part, and in its deeper portion lies behind the ramus of the mandible. So it consists of two parts, superficial and deep part. The parotid gland weighs about 14 to 28 grams and is intimately associated with the peripheral branches of the facial nerve. The duct of the parotid gland is called the Stenson's duct, and it runs forwards across the masseter muscle, turns inwards at the anterior border of the masseter, and opens into the oral cavity at a papilla opposite to the maxillary second molar. A small amount of parotid tissue occasionally forms as an accessory gland associated with the Stenson's duct, just anterior to the superficial portion. The parotid receives its blood supply from the branches of the external carotid artery as they pass through the gland. The parasympathetic nerve supply to the parotid gland is mainly by the glossopharyngeal nerve. This is the anatomy of the gland. You can see the superficial portion of the parotid gland, and the deep portion lies behind the ramus of the mandible. This is the Stenson's duct arising from the anterior border of the top part of the gland. The submandibular gland is situated in the posterior part of the floor of the mouth, adjacent to the medial aspect of mandible and wrapping around the posterior border of the mylohyoid muscle. The gland has a mass between 10 to 15 grams. The duct of the submandibular gland is called the Wharton's duct, and it runs forwards above the mylohyoid muscle and opens into the mouth beneath the tongue at the sublingual caruncle lateral to the lingual tree. The submandibular gland receives its blood supply from the facial and the lingual arteries. The parasympathetic nerve supply is derived mainly from the facial nerve, reaching the gland to the lingual nerve and the submandibular ganglia. This is the submandibular gland, the submandibular duct, Wharton's duct. Sublingual gland is the smallest of the paired major salivary glands, and it weighs approximately 2 grams. The gland is located in the anterior part of the floor of the mouth between the mucosa and the mylohyoid muscle. Its secretions enter the oral cavity through a series of small ducts. They are called the ducts of Ravenous, and they open along the sublingual fold and often through a larger duct, which is called the Bartholin's duct, that open with the submandibular duct at the sublingual caruncle. The sublingual gland receives its blood supply from the sublingual and submental arteries. The facial nerve provides the parasympathetic innervation of the sublingual gland, also via the lingual nerve and the submandibular ganglia. The minor salivary glands are estimated to be between 600 to 1000 in number, and they exist as small discrete aggregates of secretory tissue present in the submucosa throughout most of the oral cavity. The only places they are not found are the gingiva and the anterior part of the hard palate. They are predominantly mucous glands, and except for the lingual serous glands, which are the von Eppner glands, they are located in the tongue and open into troughs surrounding the circumvallate papillae on the dorsum of the tongue and at the foliate papillae on the sides of the tongue. Development of the salivary glands. Individual salivary glands arise as a proliferation of the oral epithelium, forming a focal thickening that grows into the underlying ectomesenchyme. Continued growth results in the formation of small bud that is connected to the surface by a trailing cord of epithelial cells with the mesenchymal cells condensing around the bud. Clefts develop in the bud, forming 2 or more new buds, and the continuation of this process is called branching morphogenesis, which produces successive generation of buds and a hierarchical ramification of the gland. Many factors control the location of the branch points and overall structure of the gland. So the first stage in the formation of gland is called the bud formation. In the first stage of development, the underlying mesenchyma, including the overlying, induces the overlying epithelium to proliferate. So the epithelium thus forms a bud. A mesenchymal condensation is seen below the bud, right here. This is the mesenchymal condensation, and this is the bud formation. You can see the proliferation of the cells. A thin layer of basal lamina separates the bud from the underlying mesenchyme. Stage 2 is also called as the formation and growth of the epithelial cord. The epithelial bud, which is formed, proliferates to form solid cord of cells. The underlying mesenchymal condensation also proliferates. You can see the cord of cells by the proliferation of the cells in the bud. The basal lamina is believed to play a role in influencing the morphogenesis and differentiation of the salivary glands. Stage 3 is initiation of the branching in terminal parts of the epithelial cord. The epithelial cord proliferates further and its end branches into bulbs. This stage denotes the initiation of branching. You can see the branching here, and this is going to form a bulb. Stage 4 is also called as dictomonous branching of epithelial cord and lobule formation. The terminal ends branch extensively further, forming numerous bulbs. The connective tissue below this epithelial cord forms a capsule and surrounds the entire glandular structure. A capsule is formed around and it surrounds the entire structure. You can see branching and this further branching to form the end pieces. This is the lobule formation. Stage 5 is called canalization. In the next step, the cords form a central duct. This canalization is believed to occur due to different rates of cell proliferation between the outer and the inner layer of the epithelial cord, and the fluid secretion by the duct cells, which increase the hydrostatic pressure within to form a canal. Here you can see the canal formation between the cells. In addition, during this stage, branching of the system still occurs and continues further. Stage 6 is called cytodifferentiation. In the last stage of development, the terminal secretory acini and intercalated ducts differentiate from the terminal ends of the branching tree. This is cytodifferentiation and the formation of end pieces. The structure of the salivary gland. A salivary gland consists of a series of branched ducts terminating in spherical or tubular secretory end pieces or acini. The main excretory duct, which empties into the oral cavity, divides into progressively smaller interlobar and interlobular excretory ducts that enter the lobes and lobules of the gland. The predominant intralobular ductal component is the striated duct, which plays a major role in the modification of the primary saliva, which are produced by the secretory end pieces. This is the ductal system of the salivary gland, and this is the main excretory duct, which has the largest diameter. This is the main excretory duct, and this is the striated duct, which further continues as the intercalated duct. And these are the end pieces, and these are the spherical secretory end pieces and tubular secretory end pieces. Connecting the striated ducts to the secretory end pieces are the intercalated ducts. Like we saw here, these are the striated ducts, and connecting to the end pieces is the intercalated ducts. These, which branch once or twice before joining the individual end pieces. The lumen of the end piece is continuous with that of the intercalated duct. So it is the end piece, then the intercalated duct, followed by the striated duct, and finally the excretory duct. In some glands, small extensions of the lumen, the intercellular canaliculi, are found between adjacent secretory cells. Intercellular canaliculi may extend almost to the base of the secretory cell and serve to increase the size of the secretory or the luminal surface of the cells. Now, the secretory cells are of two types, serous and mucous cells. They differ in classical histological and electron microscopic analysis and types of macromolecular components that they produce and secrete. The picture is not clear, but these are the serous cells. You can see the pyramidal shape of the cells. And the mucous cells lined by demilunes. These are the myoepithelial cells. which surround the end piece. In general, the serous cells produce proteins and glycoproteins, many of which have well-defined enzymatic, antimicrobial, calcium binding, and other activities. These proteins typically are modified by the addition of sugar residues and thus correctly are called glycoproteins. Serous glycoproteins have N-linked oligosaccharide side chains. The main product of the mucous cells are mucins, which have a protein core that is organized into specific domains and is highly substituted with sugar residues. Mucins are glycoproteins basically, but they differ from most serous cells glycoproteins in the structure of the protein core. That is, they're predominantly O-linked. And also they differ by the extent of glycosylation and their function. The serous cells, the secretory end pieces that are composed of serous cells are typically spherical and consist of 8 to 12 cells surrounding a central lumen. The cells are pyramidal in shape with a broad base adjacent to the connective tissue stroma and a narrow apex forming part of the lumen. They are pyramidal in shape. So this is a histopath picture of a serous cell. You can see the pyramidal shaped cell, the connective tissue septa dividing the gland into lobes. These are the pyramidal shaped cells. You can see here the nucleus is located close to the base, and at the top part, at the apex, which is close to the lumen, there are various secretory granules. These are the intralobular ducts, and these are the interlobular ducts formed between the lobules. These are all the serous acini. You can tell by the shape of the cells. Lumen usually has finger extensions located between the adjacent cells called intercellular canaliculi that increase the size of the luminal surface of the cell. Spherical nuclei are located basally. Occasionally, binucleated cells are seen. The secretory granules, in which the macromolecular components of saliva are stored, are present in the apical cytoplasm. You can see here, these are the secretory granules that are stored at the apex of the cell. These are the rough endoplasmic reticulum, mitochondria, Golgi apparatus present all towards the base of the nuclei. This is a round spherical nucleus close to the base. These are the communications between junctions between the two cells. Basal cytoplasm contains numerous cisternae of rough endoplasmic reticulum, which cover a large Golgi complex located just apical or lateral to the nucleus. Secretory granules of various sizes and density are present at the transface of the Golgi complex. These granules increase in density as the content condenses, eventually forming a mature secretory granules. Serous cells also contain mitochondria, lysosomes, and peroxisomes. The luminal surface, including the intercellular canaliculi, is studded with a few short microvilli. The lateral surfaces have occasional folds that interdigitate with similar processes from the adjacent cells. The basal surface is thrown into irregular folds approximately 0.5 micrometer deep that extend laterally beyond the borders of the cell to interdigitate with the folds of the adjacent cell. The folding of the cell membrane greatly increases the surface area of the cell. You can see the folds here at the base of the cell. These are the folds. This increases the surface area of the cell. Serous cells as well as the mucous cells also are joined to one another by a variety of intercellular junctions shown here. You can see the junctions. A tight junction, also called as zonula occludens, an adhering junction, zonula adherens, and a desmosome, macula adherens, form a junctional complex that separates the luminal surface from the basolateral surface of the cell. The tight junctions help to maintain these cell surface domains and regulate the passage of material from the lumen to the intercellular space and vice versa. These junctions allow the passage of small molecules between cells such as ions, metabolites, and cyclic AMPs. Coming to the mucous cells. Now, the mucous cells have a tubular configuration when cut in cross section. These tubules appear as round profiles with mucous cells surrounding a central lumen of large size than that of a serous AMPs. You can see the tubular shape. These are the mucus secreting mucus cells. And this is the lumen. The connective tissue septa dividing the gland into various lobes. Again, the interlobular duct between two lobules, and the intralobular duct. You can see the nucleus is close to the base of the cell, and the empty space which contains secretory granules filled with mucus. Mucous end pieces in the major salivary glands and some minor salivary glands have serous cells associated with them in the form of a demilune or a crescent shape covering the mucous cell at the end of the tubule. Demilunes reach the lumen of the secretions from the demilune reach the lumen of the end piece through intercellular canaliculi extending between the mucous cells at the end of the tubule. Most prominent feature of the mucous cell is the accumulation in the apical cytoplasm of, there is mucus accumulation in the secretory granules at the apical part of the cell towards the lumen, which compresses the nucleus and endoplasmic reticulum against the basal cell membrane. Large Golgi complex located mainly basal to the mass of the secretory granules is present. Small granules form at the transface of the Golgi complex, which increase in size and then join the rest of the granules, which is stored in the apical cytoplasm and then which is secreted in the duct. So this was about the serous and the mucus acini, the basic structure of the salivary gland, the development of the salivary gland. That's all for this class. In the next class, we'll learn about the formation and secretion of saliva, the components of the saliva, and the clinical consideration of the salivary gland.